Today we're going to be learning the properties of solvation based on chromatography. So today we are learning chromatography. And in the end, we're going to be generating a chromatogram. So this lab is centered around photosynthesis, but we're also going to be learning how to separate these photosynthetic pigments based on molecular mass and based on solvation properties and based on polarity and based on salt concentration. So we also have to understand what is photosynthesis. What is photosynthesis? Synthesis is build up. And photo is light. It's when the plants are able to build up their macromolecules for energy. Everybody needs energy. If you are a cell, whether a plant cell or an animal cell or bacteria cell, you need energy. This is one of the defining characteristics of life. You need water and you need energy. So the plants gotta eat too. But the way they eat is very interesting. This photosynthesis formula looks like a one line. We've been through this in the past with the Guaiacol. That Guaiacol experiment with the catalase, it was actually 2 in 1. Even though the equation was really long, it was actually 2 in 1. We had hydrogen peroxide, okay, plus catalase. It was then broken down into water plus oxygen. Then the second reaction was when Guaiacol came in and changed from colorless into amber brown. So it was H2O2 plus Guaiacol. But the catalase played the first part of the reaction. The second part of the reaction was played with Guaiacol. Same thing is here with our photosynthesis. So let's look at our equation. In photosynthesis, actually we have 2 separate reactions happening. That's why they call it the light reactions or the light phase. And then there's the dark reactions or the dark phase or the Calvin phase, named after Melvin Calvin, the botanist who discovered this very important reaction. So in photosynthesis, what happens is the plants rely on carbon dioxide, which is given off by us, plus water, and they need solar energy, sunlight, and that is going to give us water and oxygen as a byproduct. And finally, what we're doing this whole equation for, why are we doing all of this for? Why are we reacting with carbon dioxide and water for to begin with? Why do we have this? What do you think we're gonna have in the box?
The green color of the plant.
So why do we have this reaction? Why do we have carbon dioxide and water and solar energy? Why is the plant going through all of this? To get what? To get water, because the plant is so thirsty, the plant needs water.
To get the food for the plant.
To get what?
The food for the plant.
Yeah, so what's gonna be in the box? What's the food? Hamburger. Sugar, so what's the sugar? Glucose, excellent. So glucose looks like this. CH2ON multiplied by an N. This is the formula of all carbohydrates. Carbohydrates look like that. CH2O. N, 2N times an O. So, an N is on the outside. So for glucose, N is a 6. N is a 6. So 6 times carbon, that's 6 carbon, atoms, and 6 times 2n for the hydrogen, that's 12 hydrogens, and 6 times n for the O, that's 6 oxygens. That is the formula for glucose. 6 carbons, 12 hydrogens, and 6 oxygens. So this is glucose. This is glucose. C6H12 and O6. So now the plant got the food, the plant is happy, the plant is going to shunt glucose to extract energy, ATP, so it can power and sustain life. And we're going to take that oxygen, and that oxygen is going to help us for oxidative phosphorylation and for aerobic respiration. Now back to our formula. This phase that happens, that is known as the dark phase. This dark phase or dark reactions, also known as the Melvin-Calvin reactions, they are all concerned with the genesis and the making of glucose, or it's also known as carbon dioxide fixation. So we fix carbon dioxide into glucose. And then it doesn't end there. Glucose is going to be polymerized to give us starch. So glucose is a monomer. Is one bead in this long string. So it's a monosaccharide or a monomer. It's gonna give us a polymer or a polysaccharide. This polysaccharide is going to be starch. So the plant, after it gets glucose, is going to store it in the form of starch. So we're still learning this very important photosynthesis reaction. What happens in the light phase? And what happens in the dark phase, or the dark reaction, or the light reaction? And on the other side is the dark phase. So now let's break in our very important photosynthesis reaction. In the plant, you would have your chlorophyll. These are chlorophylls. These are the specialized structures responsible for the propagation of photosynthesis. And in the chlorophyll, you have circular stacks like this. These stacks are actually membranous. This is the membrane, just like the plasma membrane. They look like that. And there are protein complexes. And photosystems. There is photosystem 2 that comes before photosystem 1. We have photosystem 2 and photosystem 1, and proteins in the middle known as the transport proteins. So what happens is, if you project sunlight to the chlorophylls, the chlorophylls are going to absorb. Light is a mixture of many different colors, of many different wavelengths. So we have, this is our electromagnetic spectrum. There are colors that have very long wavelength. So if you go in this direction, you see the wavelength is really long. If you go in that direction, so that's increasing wavelength. And on the other direction, that is increasing energy. This is increasing energy, and this is increasing wavelength. So when you project sunlight, all of the color spectrum is found within the sunlight, but is divided into different colors. So we have red, orange, yellow, green, blue, violet. So violet has the most energy, and it has the highest energy and the least wavelength. And red has the highest wavelength and the least energy. But what you need to know is when we project this light into the chlorophyll is not going to absorb all of the light. It's going to absorb certain particular wavelength, and it's going to allow the passage of the other wavelength. When it absorbs that wavelength, what's going to happen to the electrons? They're going to start jumping up and down. They're going to start jumping up and down, like the people in the concert. You know, the people, when they hear the music, they start jumping up and down. But the electron is not when they hear the music. It's when you emit light to them. So if we have a concentration, spinach concentration, and we boil it, and we add acetone to it, put it in the blender, chop it up, and then we put it on top of napkin, for example, or chromatography paper, squeeze it, and all the extract comes out. We have the extract. If you project sunlight to that extract, that green extract is no longer going to look green. It's going to fluoresce, and it's going to look red. Why is that? Why did it lose its color? Because it absorbed light now. Before it wasn't absorbing, but when you project the light to it, it's going to absorb color, and it's going to reflect a different color. So the plants, they look yellow and green because they reflect different colors, but they absorb colors that are different than the colors that they reflect. So for example, our plants, so now what's going to happen is when you project light to the chlorophylls, what's going to happen is the electrons are going to jump up. They're going to move up in their valence shells. When they move up, that X-ray energy, after they reach the terminal highest level, they're going to fall back down. When they fall back down, then they're going to emit a color that's different. So for example, our plants are usually green and yellow because when you project light to them, the ones who project yellow color, the ones who are yellow color, they absorb red color. And the ones who are green colored, they have actually absorbed blue color. So they absorb a different wavelength and they project a wavelength that is, so they do not absorb green and then project green. That's wrong. So when we project that white color or light, when we project light energy to our plants, they're going to look red because they have absorbed a color of a different wavelength in our magnetic spectrum, and then they emit another one. So this is happening in the chlorophyll. The chlorophyll would take carbon dioxide and water, and this solar energy is going to help the chlorophyll cleave and break down the water. It's going to break down and oxidize the water, and the water has one oxygen, so the electrons are going to start jumping up and down. When they start jumping up and down, this is happening here in the photosystem on our thylakoid. So we have a thylakoid that has all these systems, and when the electrons move up, that is going to help the photosystem send in the opposite direction hydrogen protons, hydrogen protons, hydrogen protons. When these hydrogen protons are moving down concentration gradient, they create a proton motive force. A proton motive force. This proton motive force is going to spin for me a rotor. a rotor known as ATP synthase. This ATP synthase is gonna give me ATP, and now that's how the plant gets the ATP. So that's one part. The other part is there's a stroma. This stroma is going to accept all of the electrons that have gained energy, and it will send it back into the thylakoid, replacing NADP, who has been oxidized, giving it that electron, and now we will turn from NADP plus into NADPH. That is happening on the light phase, using sunlight energy to oxidize the water off the electrons off water, and then the electrons are going to get electromagnetic energy that are absorbed by your chlorophylls, and then the electrons are going to jump up to higher valence electron levels, and when they jump up, then they will return back to their original state, emitting a different light in their light spectrum. So what we're going to do in the lab, we're going to take those photosynthetic pigments. Your plant has multiple different photosynthetic pigments. They have accessory and they have original. So the accessory chlorophyll pigments are chlorophyll A and chlorophyll B. These are the original chlorophyll A and chlorophyll B. These are the ones who majorly absorb a particular wavelength. But there are also carotenes. These are an example of accessory pigments. What do the accessory pigments do? They support the main chlorophyll A and chlorophyll B by giving them more energy, because these accessory pigments are able to absorb a much different spectrum of the wavelength that these two cannot. So the plant cannot only rely on chlorophyll A and chlorophyll B, the original pigments. The plant needs accessory pigments who are able to absorb a different variety of wavelengths that the original chlorophyll A and chlorophyll B couldn't. And that will add more energy. But the problem is, these carotenes, these accessory pigments, structurally they do not look the same. And atomically, structurally, they are not similar to chlorophyll A or chlorophyll B. So today we're going to take the spinach extract. This spinach extract, they blended spinach and added ethanol to it. And then they have filtered out the extract. And when they filtered out the extract, we're going to take chromatography paper. Chromatography paper is our medium. Chromatography paper is our medium. It is made up of cellulose fibers. Chromatography paper is our medium, is made up of cellulose fibers. And what we're going to do is each group is going to take one slice of chromatography paper. You're going to cut it up with the scissors. And when you cut it up, you're going to make sure that it's going to fit in our tubes like this. And from the bottom, you ensure you measure approximately 1 centimeter. Approximately 1 centimeter. And that is where you're going to mash and put in all of the spinach extract. This spinach extract, what does it have? It has chlorophyll A, it has chlorophyll B, it has beta carotenes, it has cyanocobalamin, it has all of the different pigments for the photosynthesis, the original ones and the accessory ones. But you cannot separate them. Now we're going to try to separate them. We're going to put our sample here. That's our sample. That's our sample. That's our spinach extract. And we're going to put our solvent in the bottom. We're going to put our solvent here. Our solvent is 9-to-1 petroleum ether and ethanol. And this solvent, whenever you put it in touch with this chromatography paper, what's going to happen to the solvent? The solute, this is my solute, this spinach extract is going to absorb and attach to the cellulose fibers that make up this chromatography paper. And the solvent is going to start moving upward. It's going to start moving upward until it touches our spinach extract. Then the solvent is going to continue moving up, and our pigments are going to start separating based on molecular mass, based on size. So the smaller pigments are going to move very easily and very rapidly, and they're going to move to the front, while the heaviest and the densest pigments are going to stay behind. They're going to stay in the back. Then, also we know that the petroleum ether is what kind of solvent? Is it polar solvent? Is it a polar solvent or a non-polar solvent? Petroleum ether.
No.
That's non-polar. So that means it will only dissolve who? Non-polar. So if it's non-polar, it's only going to dissolve non-polar substance. If it's polar, it will only dissolve polar. If you're non-polar, you can never dissolve in a polar substance. If you're polar substance, you can never dissolve in non-polar substance. So likes dissolve like. So when you look at the separation, you will see something, and then you have to make an illustration, and this is your chromatogram. Chromatogram. This chromatogram is going to have the big dot where you put the first extract sample, and then you will see some separation. You'll see a pigmentation here, and a pigmentation here, and a pigmentation here. Then when your solvent reaches the top, you're going to open your flask and get out the chromatography paper, and you're going to dry it. Then you're going to calculate the RF. The RF is the distance traveled by the pigment, divided by the distance traveled by the solvent. So you're going to measure with a ruler. If the solvent goes up 5 centimeters, so that's 5 centimeters in the bottom, and you have chlorophyll A, chlorophyll B, and you have carotene, for example. Then you measure for A what's the distance that A traveled from its initial site. 1 centimeter, the RF for chlorophyll A will be 1 centimeter divided by 5 centimeters. And the same thing for B. You're going to measure from where it was to where it has ended, and divided by the distance traveled by the solvent, and that will be the RF for chlorophyll B. And the RF for chlorophyll C is going to be the distance traveled by the pigment chlorophyll C divided by the distance traveled by the solvent, and that